they will know something about that too. It's quite nice. Okay, so today we have Dr. Brendan Chua talking to us about vaccines, vaccine development specifically. So this is where you take an idea for a vaccine and go through the whole developmental process, which you'll find is quite costly and time-consuming and labour-intensive. Brendan's another graduate from our department, so he did a PhD here and is still here working on these projects and putting things that are starting to be used in the clinic, bringing them through the whole development process. Okay, so I'll let him tell you about it.
Thanks, Louise. Good afternoon, everybody. My topic today is on vaccine research and development, and it's not going to be too sciencey, but what I will focus on is more on the applied and the translational aspects of things. So what are the important things that you need to think about and to consider to be able to take an idea for a vaccine from the seed stage right through to preclinical development and eventually clinical trials. Now, we all should be aware that vaccination has long been considered, and it still remains one of the most economically effective and successful strategies to significantly reduce mortality and morbidity rates that are caused by many common infectious and sometimes lethal diseases. And this is perhaps best exemplified by the worldwide eradication of smallpox, with polio hopefully soon to follow. Now, this table here nicely depicts the success that vaccine has had in reducing number of disease incidences in industrialized countries such as the United States. And in particular, we've seen diseases like measles, mumps, and rubella, childhood diseases, which have resulted in about a 90 to 100% decrease in incidences as a result of the implementation of successful vaccination programs. But despite these successes, the reality is that there will always be diseases that we need vaccines for, and it's just a matter of priority. And these are but a few examples of diseases that have been highlighted by the WHO as urgently needing vaccine development for. And as you can see, there are a lot of viral diseases, particularly those that have a chronic and a latent stage, but we're also starting to see the rise of parasitic diseases, in particular malaria. So how do vaccines act against disease? Well, this is quite basic, but I'm sure we're all familiar with the concept of stimulating immunological memory, where a priming dose of antigen will equip the immune system to respond rapidly and quickly and better upon subsequent exposure to that same antigen. A vaccine doesn't necessarily need to protect against infection, although, of course, it would be a tremendous advantage if it could do that. But what it should at least be able to do is to reduce disease severity and ultimately also transmission throughout the population, which leads to the establishment of herd immunity, wherein mass vaccination can protect individuals within that community that are not being vaccinated. This, however, also has limitations in that it's generally only effective against diseases that are transmitted from human to humans, so not really very good for infections that have a zoonotic origin. And you also need about 95% of the population to be vaccinated in order for herd immunity to work the best. Now, prior to the 1980s, many of the empirically derived vaccines were based on using whole inactivated or live attenuated organisms. Now, notwithstanding the fact that these vaccines have been tremendously successful in reducing disease incidences throughout history and throughout their use, however, can sometimes be associated with some risk to safety. Now, whether this be unwanted inflammatory responses or potential for an attenuated vaccine strain to revert back to its pathogenic form, or sometimes an incomplete inactivation of the pathogen itself. But nowadays, however, a lot of these issues can be addressed to some extent, but in the last few decades, we've seen this move towards rationally designed developmental strategies, and that is mainly using components of these pathogens, eliminating any virulent constituents. Now, this is obviously a lot more safer, but the flip side of the coin is that these antigens often lack any inherent features about them that make them attractive to the immune system. So more than often an adjuvant needs to be used. Now, the past 100 years or so, we have since seen the use of aluminium-based adjuvants, which have got a pretty good safety record and quite good at inducing antibody responses, but they're not generally associated with inducing good cell-mediated immunity. But in the last few decades, we've started to see the rise in the development of other types of adjuvants which can hopefully address this need. So what does it take to develop a vaccine this day and age? Well, from an academic perspective, knowing how a vaccine works, how immune responses are induced, and what type of protective responses are needed against a particular pathogen forms essentially the basis of a rational vaccine strategy. But from a vaccine manufacturer's perspective, it goes well beyond that. Now, it's estimated it can take about 9 to 14 years or so for a vaccine candidate to successfully make it into the clinic. Now, the public often laments the fact that vaccines cost so much to develop, and eventually when they make it into the clinic, they're also very expensive, and it takes such a long time too, which leads them to assume that huge profit margins are being made without really understanding the underlying economics that drive this development program, and also the costs that are involved. So a typical vaccine development cycle can be separated into 4 distinct stages. The first of which is the research phase, and this is often where the largest amount of time is spent. And the whole aim of this phase is really to identify technologies and ways to induce immune responses against a target disease. And this may involve evaluating the efficacies of these candidates in relevant disease models, defining their mechanisms of action, what type of responses that they induce. And the bottom line is that you're looking to establish proof of concept that your idea is going to work. It's also the stage where the costs are at their lowest and can often be supported by grant funding schemes. Now, it's when you start to move up into preclinical development where the costs start to escalate. And the whole idea behind this stage is that you're transitioning your candidate from research phase and preparing it to enter clinical trials. And a lot of this work will be looking at how to best formulate your vaccine candidate, its stability, how to store it properly, and most importantly, whether or not it has any toxic effects. At this point, it's probably a good idea if you can engage a collaboration or a partnership with a commercial entity to help take on some of these costs. And some of it may involve licensing of intellectual property if it's a new technology that hasn't been used before, so that your industrial partner is able to evaluate its use for their own purposes. Now, when you enter clinical development, this is when the costs really start to grow exponentially. And there are 4 distinct types of clinical trials, ranging from 1 to 4. Phase 1 looks at safety. Sample sizes are usually quite small. Phase 2 starts to look at efficacy, and your sample size will start to increase. And then phase 3 and phase 4 again look at efficacy, but in the target population and perhaps extending it to other cohorts within the population. And all these trials are very important in order to ensure that your end result is not only going to be efficacious, but most importantly, safe. And this is really where investment from vaccine manufacturers or pharmaceutical companies, colloquially known as big pharma, is needed. And as you move through phase 3 and phase 4 trials, as the results start to look good, the vaccine manufacturer might start to think about ways of upscaling the manufacture of this vaccine, and it may mean implementing new production processes or even building new manufacturing plants, further adding to the costs. So all up, it's not actually surprising to hear costs of around 200 to 500 million US dollars being put into developing a vaccine. And that's not even taking into account the vaccine attempts the developmental attempts that have failed. So it's estimated by the time you get to phase 3 or 4, about 1 in 4 of these vaccines will only make it to the clinic. So in reality, you've got 3 other vaccine strategies that haven't worked at all. So your cost really is around $1 billion for one of these vaccines to make it into the clinic. And the bottom line is that Big Pharma takes a big financial risk in putting all this money into developing a vaccine. So it needs to obviously try to recoup these costs and also to make a bit of a profit margin in order to pump it back into developing new vaccines. So what are the things that a researcher or a vaccine manufacturer needs to think about when thinking about embarking on a rational vaccine developmental strategy? Well, the initial thing is obviously to identify what you're trying to target, and that is determining the impact that a particular disease or a health problem that's caused by the disease has in a specific region or a particular community within the population. And this is going to be dependent on several factors: the frequency of the disease, what causes it, its virulence, as well as how it transmits through the population, how this disease affects the quality of life in terms of morbidity and mortality rates, whether or not the affected populations have access to adequate healthcare resources, and ultimately the financial costs to the government and the economy in terms of loss of productivity or perhaps expenses incurred trying to help alleviate this condition. And all this can be expressed as a unit known as quality adjusted life years, which essentially is the loss of years of an average lifespan as a result of contracting or being affected by this disease. And this is going to be different between developing and developed countries due to differences in sanitation, socioeconomic factors, as well as standards of healthcare. And the bottom line is all this information helps to define what the medical need is, and whether or not it's going to be cost effective to implement or to develop a vaccine, and also the target populations and individuals that will gain the most value out of the introduction of a vaccination program. So once you've identified your disease, the next thing you need to think about is how are we going to induce an immune response against it. And this comes down to the antigens that you will be using in your vaccine. Now, an antigen for a vaccine obviously needs to be able to stimulate an immune response that is at least protective or that will lead to cure or control of the disease. Antigen expression is going to be also dependent on the strain of the pathogen that you're trying to target, whether or not it's limited to just one subtype or conserved across many different strains. Where and when the antigen is expressed is also important. So which part of the infectious cycle of a pathogen that it's expressed in. So malaria, for example, is a very complex parasite, and its expression of antigens can be different as it undergoes different replication stages throughout the body. And so the location and where this antigen is also going to be very important. So for example, if an antigen is expressed on a pathogen itself or perhaps on a toxin that it produces, then an antibody response might be the best way to go to neutralize it. Or alternatively, if it's expressed on the surface of an infected cell, then antibody-dependent cellular cytotoxicity mechanisms may be involved, or perhaps also robust cell-mediated immunity is what you need. And this is where a lot of work looking at correlates of protection is very important, because it's going to inform you how best to target your immune response against the pathogen. Now, in the past, the selection of antigen using empirical methods have been largely limited by the technologies and the methods that are used. However, in this era where we're now able to sequence the entire genome of a pathogen, it's completely changed the situation, and we can employ novel strategies like reverse vaccinology to help us with this. An example of how reverse vaccinology has been used to develop a vaccine is probably best exemplified with the development of the meningococcal B vaccine. Now, meningococcal disease is quite a severe disease that's caused by the bacteria Neisseria meningitis. Systemic infection can lead to internal bleeding. It can also spread to the brain and affect the central nervous system. So it's quite a debilitating disease. And even with the use of antibiotics, it's estimated that 1 in 10 cases is fatal. And in 3 out of 10 can lead to some kind of permanent disfigurement. Now, there are 5 serotypes which are known to virtually cause all cases in humans. And we have vaccines for serotypes A, C, Y, and W. And these vaccines largely induce antibody responses that can target the polysaccharide antigens that are found in the outer membrane capsule of the bacteria. Now, these polysaccharides can be easily isolated from grown bacteria in culture, purified, and then linked to what's called a carrier protein, usually in the form of an existing vaccine like Diptox or tetanus toxoid. And the function of the carrier protein is really to help induce CD4 T cell responses. So these vaccines are called glycoconjugates. The same strategy, however, cannot be applied to generating a vaccine for serotype B. And the reason for this is that because the polysaccharide antigen that's found on this particular serotype shares homology with proteins that are found or expressed by human cells. So to get around this, what researchers did was to sequence the Neisseria meningitis serotype B genome, and by in silico prediction, they were able to interrogate the genome to look for genes that encoded for proteins that were expressed on the surface of the bacteria, that didn't share any homology with any known human proteins, but were also conserved across the different serotype B strains. And out of this, they were able to identify about 600 potential candidates. They were able to express about 350 of them in bacteria, purified them, and used them to immunize mice to generate anti-sera. Now, the antibodies in this sera were then tested for their ability to cross-react and bind to their target antigens, as well as whether or not they exhibited bactericidal activity in the presence of complement killing assays. And so from this 600 list of genes, they whittled it down to eventually 3 candidates, which were then used to form the vaccine known as MIIM30003, which is the first genome-derived vaccine that's been licensed for use, and it's currently in clinics in Australia, the UK, Canada, as well as the USA, as well as some countries in Europe too. So it's a very, very effective strategy to look for antigens that have yet to be discovered, and currently it's being used also to identify antigens from other types of bacteria, in particular antibiotic-resistant pathogens. So now you know what you're going to target with your immune response. The next thing you have to figure out is how are you going to induce an immune response? Now, many of the empirically derived formulations were largely based on using live attenuated microorganisms, and these were great vaccines because they mimic natural infection without causing disease, and the types of immune responses that they usually induce is very well suited against the original pathogenic form. The limitations, however, are that not every microorganism can be cultivated in the lab and grown in culture, and their use also poses some risk to those who are immunocompromised, and the use of these vaccines can be akin to a natural infection with the pathogen because of their impairments in their immune systems. And then there's also the possibility that many of these attenuated organisms could revert back to their pathogenic form. And in fact, this is what happened with the Sabin polio vaccine in the US during the 1970s to 1990s, where even though it was a very rare occurrence, it occurred in about 1 in every million doses, it was still enough for the disease to persist in the population. And so since then, this vaccine has been replaced with an inactivated Salk vaccine, which had a few teething issues in itself, which I'll talk about a bit later on. Now, in the past, attenuation of these microorganisms were somewhat very rudimentary and was achieved by simply passaging the microorganism either through cell lines or through animals until they became attenuated. And often they didn't even know where these mutations had occurred before using them as a vaccine. However, in this day and age, we can again employ a lot of genomics-based approaches to try to selectively delete or mutate the genes that encode for virulence or pathogenic factors. Now, deletion is obviously the much better option because you're eliminating then the possibility of reversion occurring, but you also have to remember that and hope that this deletion doesn't affect the fitness of the pathogen. So it still needs to be able to generate the immune responses that you need. Rational modifications have also now allowed us to optimize the growth of these microorganisms for manufacturing. So for example, the influenza inactivated vaccine is created by reassorting a high growth strain with a strain that expresses the antigens that you want. Now, a much safer way to use these organisms as vaccines is by literally either killing them or inactivating them so that they can't replicate or infect anymore. And this can be achieved by using chemicals or exposure to high temperatures or even radiation. And the beauty about this approach is that the antigens that are expressed on these organisms are usually retained in the right conformation, so they can still induce the responses that will cross-protect against the actual live organism. Some limitations include the fact that because they're not replicating, you don't get this continuous production of antigen. More than often, boosters will be required. And there is also some risk that there is a carryover of components that can cause unnecessary inflammation, although in this day and age, the methods to remove impurities from these vaccines are usually quite thorough and sophisticated. But then there's also this issue about getting the attenuation or the use of the inactivating agent. It's sort of a balancing act. So too much of an inactivating agent can destabilize or degrade your antigen, which affects then your vaccine efficacy. But too little of it, then you run the risk of a live organism slipping through and being quite catastrophic, which is what actually happened back in 1955 in a situation known as the Cutter incident, named for the laboratory that it happened in. And what happened was that incomplete inactivation of the polio vaccine actually resulted in 40,000 cases of polio, leading to 10 deaths and hundreds of cases of paralysis. And this situation really paved the way for establishing very stringent, federally regulated safety standards that we know of today for the development of vaccines. And taking it a step further down in terms of safety, we're starting to see manufacturers now move towards using subunit-based formulations where you literally just use the antigens that you need without having any virulent components incorporated. Now, the advantage is that not only is it a lot safer, but it's also very easy to characterize biochemically and biologically. And because they're usually made using DNA recombinant technology, the scaling up of these antigens can be quite straightforward. Limitations are that, of course, you need to know what the antigen that's going to give you that protective response. But like I said before, with reverse vaccinology, this has helped solve many of these problems. And as I mentioned before also, that because these antigens alone don't have the ability to stimulate the immune response on their own, you need to use boosters, or in many cases, an adjuvant is required to maintain immunity throughout the lifespan of an individual. At the moment, many of these vaccines are geared mainly to induce antibody-mediated immunity. So they're very good against pathogens with low antigenic variability. But hopefully, with the rise in the development of new adjuvants, we'll start to see them also being used to induce cell-mediated immunity. Subunit-based vaccines can take on many forms. They can either be proteins or as simple as peptide epitopes, which represent sequences that antibodies and CD8 T cells can recognize. They can also be incorporated as virus-like particles, like the Gardasil vaccine for human papillomavirus, or toxoid components like diphtheria and tetanus. And we've heard also with the meningococcal B vaccine, they can be incorporated as glycoconjugate vaccines. Now, the newest kids on the block with regards to vaccines are nucleic acid-based formulations, which at its most basic form is literally a DNA which encodes for the antigen that you need. And the way it's meant to work is that you inject this DNA into the muscle, and the cells within the injection site will take up this DNA and then translate it to produce the antigens that you need. So it's very simple in composition and can be relatively inexpensive to manufacture. But the downside is, unfortunately, a lot of the proof of concept results that we've seen in many animal trials have not translated well into humans. And there is a general assumption that this is attributed to poor delivery of the nucleic acids into the cells, and also a lack of immunostimulation. Where they have seen efficacy, however, is in so-called prime boost regimens, where the DNA is used as a priming dose, followed sometime later on with the actual protein version of that antigen. So it's likely that the next generation of these vaccines will have to incorporate some level of, some way of getting this DNA into the cells. And electroporation could be a technique, and this involves implementing an electrical field to temporarily allow the cell membrane to destabilize and become more permeable so that the nucleic acids can get in. And also maybe incorporating some genes that encode for immunostimulatory molecules. What we're seeing more recently are RNA being used as vaccine templates. And the beauty about using RNA is that it doesn't have to get into the nucleus to be translated. It can all be done in the cytoplasm. And some RNA intermediates can also act as signals for pattern recognition receptors, so they may have their own self-adjuvanting effect. Now, once you've worked out how to best induce an immune response, you need to start thinking about how to deliver these vaccines. Now, in the case of attenuated microorganisms, this isn't so much of an issue because by virtue of their ability to infect and replicate cells, by nature, they can deliver themselves. And in fact, many types of viruses have been used in this way as vectors to carry antigens from other types of pathogens. And there's a whole different range of vectors that can be used. Each, of course, has their own pros and cons. And many of them have also been tested in clinical trials against many diseases. But unfortunately, none has yet to be licensed for use in humans. And there are several reasons for this. One is the presence of pre-existing anti-vector immunity, which means that if you use it a few times, eventually it will lose its efficacy. Some vectors are also very difficult to grow into manufacturing levels, but I think the underlying concern to do with these vaccines has to do with safety. And this is in light of a trial that Merck conducted in 2004 using an adenoviral vector construct carrying HIV antigens. And what they found was that vaccination actually increased susceptibility to HIV infection. And up to this day, they don't know why it's happened, but there is a suggestion that it could be the vector itself is inducing or activating vector-specific CD4 T cells, which makes them more vulnerable to HIV infection. But nonetheless, we have started to see a resurgence in the use of these delivery systems again, and this has been prompted by the development of new serotypes of viruses to get around the issue of anti-vector immunity. Encouraging results have also been demonstrated in a recent trial using a vector to introduce antigens from prostate cancer. And the results from this trial have been quite exciting in the sense that we can actually extend the survival rate of men with terminal prostate cancer by up to 40%, so it's something to keep an eye on. Now, in the case of inactivated or subunit-based vaccines, because they lack any features about them that make them attractive to the immune system, there needs to be some sort of way to I guess, make them seen by cells of the immune system. And one way is to incorporate components that can be recognized by pathogen or damage-associated molecular patterns. And the whole idea is that these components can be recognized by dendritic cells. It results in their activation and then enables them to migrate to draining lymph nodes to interact with naive T cells. So based on this very oversimplified representation of immune induction, we can see that the dendritic cell is the cell that you really want to target with a vaccine to give your vaccine every chance it has to induce an appropriate immune response. And this is where adjuvants come into it. Now, I mentioned aluminium-based adjuvants, but there are also now 3 other adjuvants, MF59 and proprietary AS adjuvants that have been developed. And the advantages are that, of course, they can improve vaccine immunogenicity. They can also induce what's called dose-sparing responses, where you don't have to use as much vaccine as you would normally need. The disadvantage is that sometimes they can cause unwanted inflammation, and it can also add complexity to the manufacturing process. So it increases the amount of, I guess, regulation involved in approving these sorts of vaccines. Now, each adjuvant has a unique immunological signature in that more than often they can induce very different types of immune responses, and it really comes down to differences on how they work, so the mechanisms of action. So you can't assume that just because an adjuvant works for a particular vaccine, that it's also going to do the same thing with another different vaccine. And this is why evaluations or trials need to be carried out using an adjuvant that is specific for one particular vaccine. Now, adjuvants themselves have been developed into much more refined and safer forms by using components that, for example, agonists that target specific pattern recognition receptors. Now, prime examples of this, and you've probably heard this in many of your other lectures, are agonists that can activate or engage toll-like receptors, both the extracellular receptors as well as the intracellular receptors. And then you also have C-type lectin receptors that can be targeted, and also these intracellular sensors like your RIG-I-like receptors and NOD-like receptors. Now, the idea to use these agonists, target vaccines, has been around for a while, and we've started to see results now coming out from clinical trials that have used, in particular, toll-like receptor agonists against a range of different infectious diseases, including cancer. Similar approaches have also been shown to work against C-type lectin receptors, and we're now also starting to see the development of agonists that can target these NOD-like and RIG-I-like receptors, although they're still in their experimental stage. So to evaluate vaccine immunogenicity, there are various parameters that you can measure depending on what your vaccine is meant to do and what it's trying to target. Now, if it's a vaccine that's geared towards inducing humoral responses, normally the assays used here will be focused on looking at the specificity of the antibody, how much you are inducing, and also the isotypes, or the profiles of these antibody responses. So, for example, you'd be looking for an IgM to IgG switch, or perhaps even the presence of secretory IgA, which is secreted as a result of mucosal immune responses. And then there are also sophisticated technologies now that enable us to measure the avidity and the affinity of binding between an antigen and an antibody. Now, with regards to cell-mediated responses, a lot of these assays are focused on looking at the function and the phenotype of the T cells that are induced. And we can look at things like their frequency, the cytokines that they produce, namely interferon gamma, IL-2, and TNF-alpha, their ability to kill pathogen-infected cells, and of course, their subclasses and the phenotypes of the T cells that are involved, and their differentiation state. Also important is looking at innate immune responses. And the reason for this is that it can often give you an indication of just how toxic or how inflammatory your vaccine approach is going to be. And in this situation, you want to be looking at infiltrating innate cells, cytokines that are produced, such as IL-6. And another really good parameter to look at is the effect of a vaccine on dendritic cells. So you can look at things like how well it activates these cells, what cytokines that are produced, and also what types of the level of antigen presentation that these cells are capable of. But ultimately, the bottom line is that these responses need to translate to some kind of biological efficacy. And to do this, it's often done in small animal model species like mice or perhaps even non-human primates, which involves challenging these animal models with the target pathogen strain that causes the disease. And you want to be using the model that best represents or captures the disease. Then you also have to think about how you're going to use your vaccine. Is it as a prophylactic or perhaps as a therapeutic vaccine, in which case you need to establish infection before vaccination. Now, let's get to the business side of things. Now, if a vaccine is new and it hasn't been used or seen before, then you need to think about how to protect this intellectual property, and this is through the use of patents. Now, patents are essentially a monopoly right that's granted for an invention to the owners or the creators for a set period of time, which means no one else is able to exploit that technology or that idea for commercial benefit except the owners of the patent. Patents are held usually for 15 to 20 years, in which case after it expires, anyone else can come in and reproduce that technology for their own purposes. So the idea is that it doesn't stifle research and development. And it's an important system because it offers recognition and material reward, usually financial, for the inventors. But it also means that vaccine manufacturers are able to invest all this money in there, knowing that when it does make it into the clinic, only they can exploit it and no one else can. There is strict criteria as to what can and cannot be patented, and there's usually four factors. Obviously, it has to be new. It can't have been previously described in any sort of form prior to the application. It must also be more than an obvious extension to what is already known. So the question to ask here is, if someone is able to come to this solution naturally given the information that is available, then that's an obvious solution, so it can't be patented. Virtually pretty much anything can be patented, and it could either be a process or some kind of a treatment for a particular disease. It could be a machine or a device that does something. It might also be a way to manufacture a vaccine or an improvement to current production methods. Or it could be as simple as a composition of matter pattern, which is literally two or more constituents being put together to do something. But importantly, what you're trying to patent has to be useful, and it has to do what is stipulated in the patent. And a lot of this is judged or reviewed by fellow scientists or engineers, usually with some knowledge of the legal processes. And these individuals are usually hosted by patent offices in many target jurisdictions throughout the world. So I've got a couple of good 10 or 15 minutes left, and I just want to spend this last few slides sharing with you some of our own experiences going through this whole process in the development of a novel vaccine delivery system. And this is work that we've been doing now for about 7 to 10 years. And we invented something and we patented it, and at the moment we've established proof of concept, and now we're undergoing preclinical development to try to get this new technology ready for clinical trial testing within the next 2 years. And the basis for this invention lies in its ability to target vaccines' pattern recognition receptors. Now, I said before that it's not a novel idea. There's a lot of prior art, so it's been done before. But the situation is that many of these agonists are usually formulated with an antigen, either as an admixture, where you literally just mix the two together in solution, or by having both of them conjugated together. Now, conjugates are the much more effective option, because if you think about it, if you have the antigen and the agonist together, then antigen uptake as well as activation of a dendritic cell can occur at the same time, which means that your ability to cross-prime this antigen to CD8 T cells is going to be much higher. However, the issue is that many of these agonists are non-peptidic components, in a sense that they're very difficult to use because they often require chemical modifications. They can be quite expensive as well, and prove to be quite challenging to achieve. So what we wanted to do was to try to come up with a much more easier way of conjugating these agonists to an antigen. And now at the moment, the best way to do it is what's called a covalent conjugation. But what we have tried to invent is achieving this conjugation by means of an electrostatic interaction. And the proof of concept we've demonstrated is using this particular agonist called PAM2CIS, which is recognized by toll-like receptor 2. Now, TLR2 for short is expressed on many cells of the adaptive and innate immune systems, but in particular dendritic cells. And engagement of this receptor results in cell activation, production of secretory cytokines, as well as it activates the cells. And this here is just a picture of this particular ligand. And it's a very simple fatty acid that fits into the receptor like a foot fits into a shoe. Now, to facilitate electrostatic interaction with an antigen, we've taken this PAM2CIS molecule, we've incorporated it in a peptide structure. So this is literally a structure that's made out of amino acids. So it's very simple. But a notable feature is that it has this branched structure. And at the N-terminus of the structure, we've attached arginine residues, with each residue having a very strong positive charge. And this gives the overall lipopeptide a positive charge, which then allows it to electrostatically associate with an antigen. And we can measure this interaction very easily using available chemical methods and technologies. And what we've shown is that this whole complex can effectively deliver the antigen to the dendritic cells, induce their activation, which then leads to the induction of various immune effects. And we've published extensively on the mechanisms of action, what types of immune responses that this particular delivery system can do. But perhaps the best demonstration of its proof of concept is with regards to how it can be used to improve our ability to protect against influenza. Now, I've just heard that you've been lectured on influenza, so I won't talk about it too deeply, except to say that the current influenza vaccine induces neutralizing antibodies against the hemagglutinin and the neuraminidase surface glycoproteins. And these vaccines are literally viruses that have been inactivated and then detergent split into its various components. However, as you'd know, because of the ability of this virus to rapidly acquire mutations in the genes that encode for these antigens, often the vaccine efficacy will wane over subsequent years, and then you need to get another new vaccine. So it's not very effective, particularly against an influenza strain that has drifted or one that you may encounter in a pandemic outbreak. So to look at how this delivery system works with an influenza vaccine, we've managed to get our hands on a split virus formulation. So this is a H1N1 virus called PR8, and we simply mix the vaccine with the delivery system. Now, the first thing we looked at was innate immune responses, and we did this by literally just challenging animals 1, 3, or 7 days after vaccination, took out their lungs and looked at viral titers. So what you're seeing here are lung viral titers induced by the adjuvanted formulation, which is in black. And you can see here at each time point, there are significantly lower viral titers in their lungs if you were to use this H1N1 virus. Now, if you were to use a H3N2 virus, so this is a strain that's not in the vaccine, so it's a non-matched strain, we also see this protective effect. And what this says to us is that the innate immune responses that are induced by this particular vaccine delivery system is able to protect against different strains of influenza viruses. And we have also shown that it works against other types of viruses like respiratory syncytial virus and also other bacterial strains like Legionella as well as Streptococcus pneumoniae. So what about adaptive immune responses, which is really what we're looking for? So to look at this, on day 9 after vaccination, we've taken out their lungs as well as the mediastinal lymph nodes of vaccinated animals and looked for the presence of cytokine-producing T cells. So these are T cells, CD8 T cells, which produce interferon gamma, TNF-alpha, or IL-2. And in each case here, you can see here in the black bars, the animals that have received the adjuvanted formulations, there's a significantly higher number of these cells that have been induced. Now, with regards to antibody titers, we've taken the animals out to 34 days and looked at vaccine-specific antibodies in the blood. And this result here just shows that if you go by the intranasal route or the subcutaneous route or the intramuscular route, in each case you get a vast improvement of antibody responses. And importantly, these antibodies are able to neutralize hemagglutinin binding of the vaccine matched strain, but not against the non-matched strain, which is what we expect. Now, when we challenge these animals, this is again at day 35, and look at lung viral titers, if you were to use the vaccine matched strain, we see that there's virtually no virus being detected. So it's protective. It's just neutralized the virus straight away. Now, if you look at a non-matched strain, however, we also see some kind of a protective effect, although not clearly as effective as if you were to match the vaccine strain with the challenge strain. But nonetheless, it's still quite a remarkable finding because it's showing that our delivery system is able to induce some kind of a cross-protective response. And we have since also shown that this response is mediated by cross-protective CD8 T cells. So overall, we found that the use of our new adjuvant can really help improve the magnitude of responses as well as the utility of an influenza seasonal vaccine. And it does this by being able to stimulate innate immune responses, so it gives you that short window of protection that doesn't last long, it sort of dies off after 7 days. But it can also cross-protect against different influenza strains not covered in the vaccine. So it works as an antiviral, so to speak. But then if you wait a couple of days later or a couple of weeks later, you start to see adaptive immune response start to come into effect. And this is mediated by CD8 T cells to induce cross-protective immunity as well as antibody responses. So at the moment where we're at is that we're trying to extend these findings into a ferret model because this is the more accepted model, animal model for evaluating influenza vaccines. And at the same time, we're also now looking at preclinically developing this vaccine. So looking at its stability, how it can be formulated, as well as any potential toxic effects before preparing it for use in clinical trials. So my last slide is just some overall take-home messages, and I hope I've been able to show you the reasons for why vaccine development is so long and such an expensive process. And you really have to take a look at it from the perspective of a vaccine manufacturer. It needs to do anything that is possible in order to make sure the vaccine is safe. That's your number one priority before looking at efficacy, because obviously of any potential litigations in the future. Patents are very important for vaccine manufacturers, because if they don't have exclusive use to exploit the technology, then they're not even going to bother putting any money in, because it's not going to make any business sense for them to put in these hundreds of millions of dollars to develop it. Some of the considerations for an effective vaccine strategy, need to think about what constitutes a rational approach. So think about the reasoning and the logic behind all this. To take into account also the vaccine forms that need to be used, their pros and their cons, how they work, how some of them could be very efficacious but at the expense of safety, and vice versa. And also some of the things that can be used to improve vaccine immunogenicity by facilitating enhanced delivery. And there are many different ways of doing this, so something to keep in mind. So I'll just end also by just adding some references which may give you some indication of the whole process and the current state of where we're at with vaccine development, particularly rational vaccine development. Thank you. Happy to take any questions.
In terms of the TLR2 agonist that you've developed, in the mice that have been vaccinated, I understand that some of these development of a vaccine that also has cellular involvement is CD8 immunopathology. So I was just wondering, in those mice, what was one challenge, what was the immunopathology like?
That's a really, really good question. And certainly as part of our experiments, especially with the animal ethics that we have to use, it involves monitoring these mice for any potential adverse incidences. So we look at things like, obviously redness or swelling, the behavior of these mice, whether they've changed. Weight loss is a particularly good indication as well. So what we've used with this particular delivery system is that we're using such tiny amounts that we actually don't see a lot of pathology in that sense. Obviously, if you use more, then you'll start to see it happen, but then that's, with many things, too much of anything is not good. So at the moment, it looks good. Our tox studies are looking quite good, and we've gone into rabbits and guinea pigs, so much larger animal species. The good thing about looking at larger animal species for tox is that if you don't see many toxic effects in mice, then you're not going to see it in larger animal species. If you do see it in mice, and then you go to larger animal species, they're usually they're more minimal because of the size of the target animal model that you're using. So it's looking good at the moment. Yep.
Just to clarify sort of a mechanistic point, when you're talking about conflicts between pattern recognition receptor agonists and vaccines, you mentioned the role of dendritic cells. So as I understand it, is what you're describing that the pattern recognition receptor agonist will bind and then you actually get the antigen taken up into the cell and cross-presented in MHC1 as like a receptor-mediated cytosis kind of.
Exactly. Yeah, and that depends also on the receptor that you're targeting. So for us, TLR2 behaved exactly like that. It captures antigens very well. It endocytoses and it accesses the cross-priming pathway. It accesses MHC class 2 pathways as well. So yeah, it depends on the receptor. Obviously, pattern recognition receptors on the surface of the cell is much more easier to target. Yeah, because if you're trying to target intracellular receptors, that's going to be difficult. Then you're sort of relying on the intrinsic feature of the cell to be able to take it up on its own. Now, the good thing about dendritic cells is that they macropinocytose constitutively, so they're obviously sampling their environment. So that might be one way of getting antigens into cells efficiently by that way too.
I just got a question in regards to the innate immune response. You said that it was 7 days, right? That the mice had an antiviral state for this amount of time. I was wondering how that could happen without the mice feeling this sick.
Well, we don't know whether the mice are feeling sick without asking them, and they can't respond, obviously. Like I said, all we've got to gauge is, their behavior and any other pathological symptoms that we can visually see. We know that it is an innate immune response because we do see a flooding of innate cells into the lungs. So you get a lot of neutrophils, NK cells, macrophages, which is in a way accounts for that non-specificity of the innate immune response. But the good thing is that, and what I haven't mentioned, is that these responses, if you don't put the vaccine in there, these innate immune responses eventually die down, and the mice, and you lose protection. And that's the point. So if you include the vaccine in there, once they lose that innate protection, mediated protection, then the adaptive immune response kicks in. But pathology in the lung is a big, big problem, and there are ways to get around it. I can't talk about it too much now because it is, we have got patents on this, and we need to be careful about what we say. But I know you guys are pretty good, and no, I'm not going to say anything. I just want to also mention we have a couple of honours projects going on at the moment, and we are in the midst of currently screening prospective students. So if anyone's interested, just drop me a line and email me.
Good? Okay, thanks.
No worries. That's good. 50 minutes. Is that right? Okay. It's hard because
You were concerned for no reason.
well, it's hard because